Transgenic animals. Transgenic animals. Let's see about a brief information of the transgenic animal. Animals that have their own genome and had their DNA manipulated to possess and express an extra or foreign gene are known as transgenic animals. This word is more famous nowadays. The transgenic animal that have animals. In the case of plants, it was were much advanced actually. Animals that have their own genome, they have their own genome and had the DNA manipulated to possess and express an extra foreign gene, which is introduced into that particular genome. That we say such kind of animals as transgenic animal. Okay, transgenic rats, transgenic rabbits, transgenic pigs, sheep, cows, and fish have been produced nowadays. So transgenic Jamana. Although over 95% of all existing trans animals are mice, because it's a laboratory animal, that's why we did more experiments on this particular mice itself. That means for medicine, for treatment aspects, for the various test aspects, we did more transgenic mice as we record that we created. Transgenic animals are produced for many reasons, aiming at benefiting human beings. That reasons we are learning now under four that main aspects. We are going to learn in that manner. Let us try that and explore some of the common reasons here. Let us try normal physiology and development. Laboratory experiments here. The scientists experiments for their beneficial to their experiments. These particular transgenic animals till the time is created over in four aspects. Number one, normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. The study of complex factors involved in the growth, such as insulin-like growth factor, insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects of that result. Information is obtained about the biological role of the factor in the body. Simple insulin-like growth factors will be isolated, the genes will be isolated if we inject in the mice, that if any for that additional growth or additional effects is seen over, that is the activity of insulin-like growth factor that we can understand. So that's why in the normal physiology and development. Next, study of diseases. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of diseases. In case of vaccines, also you may heard over. Stage 1 is over, stage 2 is over, now stage 3 on human beings, these kind of things. So in first stages, they do have that these kind of experiments on that transgenic animal. Many transgenic animals are designed to increase our understanding how genes contribute to the development of a disease. These are specially made to serve as a models for human diseases so that investigation of new treatments for the disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's diseases. These all disorders to discuss to test that particular disorder. Separate transgenic animals are created. Next, biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to produce. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA or gene which codes for a particular product such as human protein, very important alpha 1 antitrypsin, which is used to treat emphysema. Emphysema, did you remember a respiratory disorder? That alpha 1 antitrypsin is going to be produced from the transgenic animals only, a biological product at alpha 1 antitrypsin used to treat emphysema, a respiratory lung disorder which will come by cigarette smoking. Then similar attempts were made for treatment of phenylketonuria, alkaptonuria, cystic fibrosis, hemophilia. That all these are single gene disorders. Did you remember? Then in 1997, the first transgenic cow, one more here. This is that means total general information, but some technical words we have to remember for examination point of view. The first transgenic cow, Rosie. produced in a human protein enriched milk, 2.4 grams per liter. Okay, Rosie is produced. That first transgenic cow, Rosie, is produced with human protein enriched milk, 2.4 grams per liter. This milk, the milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than what is available in normal milk from untreated cows. Untreated means that not transgenic cow. In case of a transgenic cow, this is Rosie, that we are able to insert our gene which can produce the alpha lactalbumin protein, that and we produce a protein enriched milk. Okay, so Rosie alpha lactalbumin, 2.4 grams milk, that per milk per grams of protein per 1 liter of milk. And one more thing, alpha 1 antitrypsin, which is used to treat emphysema. These two things are very, very important. Alpha 1 antitrypsin, alpha lactalbumin. Don't confuse between them. Alpha 1 antitrypsin used to treat emphysema, and alpha lactalbumin is the protein which is there in the balance that produced in the transgenic cow. Okay, what is actually alpha lactalbumin? Is rich in essential amino acids and is the dominant protein in human milk. Actually, we are able to produce that kind of human type milk in Rosie cow also by introducing the gene in the Rosie. Next, vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines. Before they are used on humans, transgenic mice are being used to test the safety, used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of the batches of the vaccine. That's it's very, very difficult to bring out a single vaccine out. That various experiments we have to do. Then after finally on human beings, they do. A transgenic mice are being developed for use in testing the safety of vaccines. Before they are used in human, transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches. Actually, we do on monkeys, but nowadays we are doing on transgenic mice because monkey is close to human being. But extinction problems are there. That's why. Then why not mice? Mice is a very, very good laboratory animal, so that it can produce that fertility and production capacity is more for mice. That's why more laboratory experiments will be done on mice. But vaccine-like conditions, that we that monkey is very closer to the human being. That if we do on monkey, it will be very good for us actually. But the extinction problems are there. People are allowing the do on monkeys. That's why we are creating a transgenic mice, which is to look like closer to the human activity. Next, finally, chemical safety testing. This is known as toxicity or safety testing. After a medicine we identify, that will test like this. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals that carry genes which make them more sensitive to toxic substances than non-transgenic animals are produced. Okay, so that's why we are using transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. Indian government has set up that GEAC, Genetic Engineering Approval Committee, to look into misuse of DNA manipulation and safety of the introducing that genetically modified organism for public services. Otherwise, people will transfer that gene to this gene to that gene. That is good in the case of Avengers and Spider-Man, but in reality, that's not good, isn't it? Yeah, so that's it. These are all about the four aspects which we are doing in case of transgenic animal. The four major benefits. The four major benefits like normal physiology and development, study of disease and biological products, vaccine safety, and chemical safety testing. And finally, again, I'm showing here the very, very important things to be remembered in this particular case are that alpha 1 antitrypsin, that alpha 1 antitrypsin, very, very important. Alpha 1 antitrypsin and one more alpha lactalbumin. Okay, alpha lactalbumin. So these two are very important from this particular area, along with our ROSI. ROSI also to be remembered. So that means in NEET examination point of view, these three are going to be very important. Alpha 1 antitrypsin, ROSI, and alpha lactalbumin. Okay. So let us see once again what they are. Alpha 1 antitrypsin is a human protein, a product of human protein used to treat emphysema, which were produced in transgenic manner as a biological product. ROSI introduced produced human protein enriched milk 2.4 grams per liter. The milk contains human alpha lactalbumin. These two things is very important to be remembered. Okay, so that's it. That's it about transgenic animal. Transgenic animal.